It's my pleasure to introduce our speaker today, Dr. Jayanthi Nadarajan. She is the science team leader for the Germplasm Conservation Team at Plant and Food Research New Zealand and program leader for the New Zealand Ministry of Business-Funded Nationally Significant Databases and Collections. Prior to joining the Plant and Food Research in 2017, Jayanthi was affiliated with the Royal Botanical Gardens Kew, working on the Millennium Seed Bank program. She is also the current chair of the Seed Storage Committee of the International Seed Testing Association, ISTA, with responsibility to manage the program of the committee. Jayanthi has over 16 years experience in plant conservation science with particular focus in seed conservation. She advocates integrated conservation strategies, which include conservation of pollen and seed, complemented by in vitro and cryopreservation techniques to mitigate the effects of biotic and abiotic threats to endangered plants in field and wild populations. The title of her presentation today is Seed Storage Behavior. What do we know now that we didn't know 50 years ago? Welcome, Jayanthi.
Thank you, Yani, for the kind introduction and for the invitation to speak at the GROW webinar. It's a pleasure and honor to be part of this webinar. My name is Jayanthi Nadarajan. Good morning, good afternoon, evening to everyone who joined this webinar from whichever part of the world you're joining from. My talk for today is seed storage behavior, what we know now that we did not know 50 years ago. I selected this topic just to reflect on the knowledge evolution, the technology evolution in this area, that how the technology have evolved so much, or the knowledge have evolved so much that we know so much more now in seed storage science that we did not know 50 years ago. So my talk today will be part of the review paper that we published last year on seed longevity, the evolution of knowledge and conceptual framework. This is a collaborative work from my colleagues from Royal Botanic Garden Kew, USDA, and from Spanish University. So my collaborators, I would like to thank my collaborators, Christina Walters, Hugh Pritchard, Daniel, and Louise for their collaborative work in this area. So this is a vast topic. I would like to concise the talk into or reduce the scope into several subheadings. I will be concentrating on the seed moisture content that affects storage, seed storage classification, the relationship between seed moisture content and relative humidity, temperature, cytoplasmic solidification that's involving molecular motion and seed longevity. Can we predict seed storage behavior, aging of orthodox seeds during storage, some recommendation for storage, looking at different storage categories, and some take-home messages. Just to give some perspective on the history, seed storage science is not new. In 1832, Augustine Paramus has already had a chapter on seed preservation in his book, Physiologiae Vegetale, or Plant Physiology. He pointed out that vitality of seeds would be prolonged if seeds are stored under condition to protect them from heat, moisture, and oxygen. So these are the three factors that we are still investigating after 200 years. And in 1908, Everett created a list of short, medium, and long-lived seeds under ambient environments. So at that point, there's no control environment condition, but we know that seed can be stored at ambient environment, and seeds behave differently, different classes for storage behavior. And precisely 50 years ago, Robert and Alex published an article on proceedings of storage life, predicting storage life in seeds. So 50 years have moved on and we are investigating same areas, but our knowledge have gained or evolved so much that we know more. And I'll be touching some of the areas where the knowledge have evolved. Why seed storage has gained so much attention? It is the most efficient approach to ensure the long-term preservation of genetic resources. And in the last 50 years, I've seen basic questions, how seed storage can be prolonged or how seeds can be maintained for longer period. And the reliable prediction of duration for which seed can remain viable, for example, longevity or lifespan, has been a concern for more than 100 years or nearly 200 years now. And the understanding of different seed storage classification, that is a requirement, especially for either gene bank or breeders, plant breeders or farmers. So how best we can store the seeds and the understanding the factors that contributing to seed longevity to lead major advancement in seed gene banks. It's created a more interest in this area. Just before I go detail into the talk, just want to highlight the storage requirement or the objectives for industry, seed industry farmers or breeders are slightly different from gene bank. For farmers, it's usually, or industry is usually focused on economically important crop species. And they're usually looking at preserving plant materials from one growing season to next growing season without having to resow them every season. And farmers has found that the advantage of carrying over the seed storage, so that is improve the soil quality as well. Different species and varieties can be stored, and this also gives the advantage of free movement of seed commerce in the world. And also farmers or breeders have the advantage of selecting breeding lines or traits that's very useful for their production. Whereas for the gene bank, the storage or the objective for storage is for long-term conservation. This is mainly as policy in species extinction. This is for wild species, crop wild relatives, and to conserve and promote biodiversity and protecting food plant biodiversity as well. And gene banks also act as like to provide materials for research and reintroduction. There are more than 1,750 seed banks in the world, and the accessions are over like 7 million accessions being stored, and some seed banks are dedicated just to conserve wild species. We know that moisture content is the most important factor in maintaining seed viability during storage. And Harrington's rule of thumb indicate that for every 1% reduction in seed moisture content, the longevity can be doubled. At low moisture content, the seed aging is delayed because there's low metabolic rate of activities, and that prolongs longevity. But we need to be very careful not to dry, because there's a lower limit of moisture content that the seeds can withstand. Drying below that lower limit will cause damage, lethal damage to the seeds. Some seeds may withstand very low drying, sometimes up to 2% of moisture content. However, care need to be taken when rehydrating the seeds before germinating, because imbibitional damage can occur and could damage the seeds. A note of caution here is not all seeds respond to desiccation equally. So, having said that not all seeds respond to desiccation equally, now there is the opportunity to classify the seeds, how seeds respond to desiccation, and if they behave differently in storage. So seeds that do not survive desiccation are frequently referred to as recalcitrant seeds on account of difficulty in storing them. A larger proportion of species produces desiccation tolerant seeds, or termed as orthodox seeds, tend to be more amenable for dry cold storage. The orthodox versus recalcitrant delineation provides an immediate perspective or an advantage to invest in infrastructure for gene banking, because these two groups of species do require different storage environment. But what we need to understand is there's no clear cut point where these are the orthodox seeds and these are the recalcitrant seeds. So the desiccation tolerance is a continuous phenotypic trait. So sometimes we do see some classification or behavior in between these two categories. For example, there's been some reports on sub-orthodox classification, intermediate seeds, class 2 seeds. Seeds are minimally recalcitrant. So there are a group of species that fit into these categories in between. So care need to be taken that not all seeds can be either orthodox or recalcitrant only. So in 1996, Hong and Ellis have created this chart showing that in reflection to the seed bank, how seed storage classification can be banked or not banked, or what requirements they would need for storage. For example, if you look at this graph here, the A is represented by orthodox seeds. So the x-axis is the moisture content, so from high moisture content to low moisture content here, and this is the y-axis is germination. For orthodox seeds, we can dry the seeds without any effect on their germination, and they can be stored at the seed bank moisture requirement, which is around 5 to 7% moisture content. Whereas for the recalcitrant seeds, they're highly sensitive to desiccation. So they will decline in germination once we start desiccating them. So storage at seed bank is not possible for this class of seeds. So we need to look at other alternatives. For the intermediate or the C here, we can dry the seeds. They can tolerate desiccation up to a certain level, but storing at low temperature will affect the germination. So we probably will compromise some of the longevity of the seeds by storing them at the seed bank moisture content or drying them to seed bank moisture content. So these are the basic seed storage classification that was introduced in 1996. So the classification of seed storage behavior then and the effect of the moisture content bring to the research on the cytoplasmic solidification and glass formation in seeds. The glass formation, also called as vitrification, describes how fluid cytoplasm solidifies when liquid water is removed or when ice formation is prevented. This is by drying. So during desiccation, the hydrated cytoplasm will concentrate and becomes increasingly viscous, and the remaining dry components will squeeze together, become compact. The mixture begins to hold its own shape when dried to relative humidity between 50 to 25%. This is above 0 degrees C, and resisting further compaction. That is, solidifies, become glass. The initial understanding was that glasses form in cytoplasm of orthodox seeds, but not in recalcitrant seeds. So that's where we thought, like, why orthodox seeds can be stored in cold temperature by drying in cold temperature compared to desiccation sensitive seeds. However, now we know that transition from fluid to solid occurs in both desiccation tolerant and sensitive cells alike. Rather, it may be the extent of cell shrinkage that occurs before glass forms that distinguishes recalcitrant and orthodox categories. If you look at the orthodox seeds, they go through maturation drying during seed development. So at the end of maturation, they will become fully dried. They have reached high water, dry matter content. And compared to recalcitrant seeds where they'll be in fully hydrated states. So the cells stocked with dry matter content reserves, this is for orthodox seeds, are much more denser and shrink less given the same desiccation force than cells containing a lot of water content, or in this case, recalcitrant seeds. So if you look at this schematic diagram here, the undried seeds is fully intact. And then when we start desiccating the seeds, the orthodox seeds will shrink about 50% of the volume, and they will survive the drying, compared to the recalcitrant seeds, which has got high water content, which they will shrink more than 80% shrinkage, and they will not survive that drying or the. So looking at this, we know that determining the glassy state of the seeds is critical. So understanding the physical properties of water and tissue that being stored is important. And we can actually use some tools to determine if we have achieved a vitrified system in seeds that we are working with. One tool that we can use is differential scanning colorimetry that is shown here. And it provides the feasibility to quantify freezable water, non-freezable water, and if the cells have reached vitrification. So an output from a DSC is called a thermogram, which looks like this, a typical thermogram, where the x-axis is temperature and the heat flow is shown in the y-axis. When we have a load of water, a sample in fresh sample, we can program this machine or instrument to cool that sample from room temperature to sub-zero temperatures, and then bring that sample back or re-warm the sample back to room temperature. When we cool our material, ice will form when there's a lot of water or free water. And when we warm the sample back, whatever ice that will crystallize will melt. So we will see that melting peak in our sample when there's freezable water, and the amount can be quantified by the area under the peak. So when there's a lot of water, the peak will be bigger, and the peak will become smaller as we desiccate the sample. At one point, when all the free water has been removed, we'll get a stable profile saying that this sample is vitrified, or we have removed all the free water. And a DSC output also can show a glass transition state, where this shows us a change in the baseline, and it's like a step-like output that we can see from the DSA. So this will usually show what temperature a glass is forming. So it's very useful, and this will help us to decide how far we need to desiccate the samples. While DSA is very useful to predict if we have removed all the free water and if glass transition is achieved, it's also important to predict how stable is the glass that is formed, because the stability of glass will actually predict the longevity of the seeds. So if seeds are recorded to store well when they've been stored under or well below the glass transition temperature. Other equipment that we can use is dynamic mechanical analyzer. So there are some research here that I have highlighted 2 papers here where you can look into more details. And DMA is more sensitive to local motion, and it can provide the most credible information on glasses and changes in viscosity that occur in a very dry system. And that's a limitation where DSC will not able to produce this, because when the signals are too small, it may not be predicted in DSC. So there are opportunities to use DMA in this instance. So now we know that seed longevity is affected by seed moisture content, temperature, as well as relative humidity. And these factors are interrelated. So this is a representative water absorption isotherm for example, a seed with 2% lipid, or starchy seed, we'll call it. So when we look at the storage environment, the dashed line here is showing the drying condition needed to meet the FAO recommended seed storage environment. So if you're looking at storing a seed containing 2% lipid at 20% relative humidity and minus 15% 15 degrees C storage, So the seeds need to be dried to about 9.4% water content. So, and this is different for different storage temperature as well as relative humidity. So we need to look at what temperature we're drying and what relative humidity we're drying. This will be different for seeds with different lipid contents, which I'll come to later. So I've shown this diagram before. This is what I call as a basic seed storage classification. But the advanced seed storage classification now is looking like this. So now we know more on what's happening with water. So this is where the water content, the x-axis, from highly hydrated to more desiccated level here, and the y-axis is the temperature. So from room temperature up to cryogenic temperature. The basic requirement is still the same. The understanding is still the same. So we have orthodox seed here, and this is the seed bank environment. So we can store the seed orthodox seeds or desiccation tolerant seeds at seed banking environment without any problem. And this line indicates the glass transition temperature line. So if you look at the intermediate seed category here, the glass transition temperature is very, very close to the seed bank temperature. And if you remember, I mentioned that it's best to store well below the glass transition temperature for longevity. So we need to look into storing at much lower temperature for the intermediate seeds. And for recalcitrant seeds, it's really crucial to store them at very, very low water content because glass transition is occurring at much lower water content. So sometimes we can't dry the seeds to this water content, and we need to apply different cryoprotectants, again, which I'll come to it later. So now we know that different seed classes would require different storage environments. So how do we predict the storage behavior of the seeds, especially if we haven't worked with this species before? We can look at the seed physiology, seed morphology, seed desiccation sensitivity assessment, and we can look back to the ecological or habitat of seeds where they come from. There's a lot of information that's been stored or created in the seed information database by the Royal Botanical Garden Kew. There's a link here. And this site is now managed by the Society for Ecological Restoration now. So if you can search this database, if you want to look at some storage behavior of a species that of your interest, so you can search by species, genre, or family to get some idea on the species storability. So just going to the seed physiology, how we can predict the storage behavior of the seeds using the seed physiology. Orthodox seeds usually have a clear end phase of seed development and maturation before they start germinating. So usually the maturation is achieved, they're fully dried at that level, and there'll be a period where usually they go through a dormancy period before starting germination. And for the desiccation sensitive seeds, this is not always the case. So there will now be a clear end phase of seed development. And usually germination will start immediately after seed development. So they don't show any dormancy period. And also desiccation sensitive seeds are usually bigger. So a report or research done by those et al. Here showed that the seed dry mass of the desiccation sensitive seeds are bigger compared to the desiccation tolerant seeds. And the mean time to germinate is a lot quicker for desiccation sensitive seeds compared to desiccation tolerant seeds. And just watch out for the inter or intra-population maturation in heterogeneity, because having a maturation in uniformity at collection is important, especially for gene banks, for preparation of the seeds and to storing them. So when you collect a population with a mixed maturity that's shown in this photo here, it's really important to segregate them and to make a uniform population before we start drying them, because a mixture of different maturity will give you different response to desiccation and storage. So we don't want to compromise that by treating this as a one population and getting different results. The seed morphology and seed morphology also can indicate or give us some indication on the storage behavior of the seeds. Usually desiccation-sensitive seeds are fleshy, and the endosperm, they have plastic endosperm and thinner seed coats. So we can look at the seed coat ratio to the endosperm, and desiccation-sensitive seeds usually have smaller ratio compared to the desiccation-tolerant seeds. And this has been reported by Dose et al. And can be found in this reference here. And when we look at ecological habitat, there are cues that we can look at. Usually desiccation-sensitive seeds are produced in tropical and subtropical environment. These are humid environments. So this is like, for example, evergreen rainforest or semi-evergreen rainforest. We still can find desiccation sensitives in temperate zones, for example, in moist, warm temperature woodlands, but in much lower frequencies. It has been reported that nearly 50% of tropical and subtropical zone species will produce recalcitrant seeds or desiccation-sensitive seeds, compared to only like about 10% coming from the dry land. So this will give us a nice indication if you're collecting species from the wetlands, most likely they'll be desiccation-sensitive. Just to give you how even as in single species can behave differently when they're collected from different populations or from different ecological habitats. This species is Sagesia mari that we worked on, and this belongs to the Myrtaceae family. So this species were collected from three different locations or populations in New Zealand, one from the north of South Island in Knoxville, and two populations in North Highlands. And we noted that the species that collected from Manawatu regions are more desiccation tolerant compared to the other two populations. And this response is different for the year of collection as well. So it's also important to record any significant climatic events that happen, like, for example, drought or any severe climatic events that happen before the collection that might affect the desiccation sensitivity of the population of the seeds that we're collecting. So it's worth noting the point. And we can also do desiccation sensitivity assessment for the species that we haven't worked with before. So, this can be done in a simple small population where we can dry the seeds to select a moisture content between 10 to 12%, do a germination test and see if the seeds are alive or die. If they die, they probably will be recalcitrant. If most of the seeds survive, then we can further dry the seeds to 5% moisture content and then do another germination test. And if they die at that moisture content, they will be probably intermediate. And if they survive, we can store the seeds at minus 20 degrees C. This is a recommended seed bank environment for about 3 months, and then do another germination test after 3 months. And if they still survive, that they probably will be orthodox. And if they die after that, they probably will be intermediate. And this is a very crude way of predicting the storage behavior. So that will give you some indication if the seeds will be bankable at minus 20 or if they need cryopreservation, for example, for long-term storage. So just to give you an example of what we did with our swarm iris seeds, this is Sagesia mari belonging to the Myrtaceae family. There's no record in literature on the seed storage behavior. So when we collected the seeds, we looked at the environmental clues and the physiology of the seeds. So they came from very humid habitat. They have fleshy seeds and very thin seed coat. So the prediction was they probably will be recalcitrant. So what we did was we did the desiccation sensitivity assessment where we dry the seeds to different moisture content. And we found that the seeds are recalcitrant because they do not tolerate to desiccation to moisture content below 20%. So this is one way of finding it. And we definitely know that these things, seeds are not storable at seed bank environment. So we need to look at cryopreservation for storing these seeds. So I'll go into the recommendation for storing seeds with different storage behavior. For orthodox or desiccation tolerant seeds, conventional seed banking is recommended. So this is storing at minus 20 degrees C after drying at 15% relative humidity. So the seeds are collected. Upon collection, they need to be dried. And usually we apply 15% relative humidity, drying at 15 degrees C. Once the seeds are dried, they'll be cleaned and checked for purity and packaged in airtight containers and stored at minus 20 or minus 18 degrees C as recommended. An interval germination test is recommended, usually every 10 years once, to see how the seeds are performing. And sometimes we'll see some seeds may require a much shorter interval depending on how well they're doing. So in theory, desiccations tolerant seeds should survive or retain their viability for a longer period. However, more data is now emerging showing that the viability is declining for the orthodox seeds that's been stored for many years. For example, her report by Lee and Pritchard, published in TIPS in 2009, showing that half-life of 276 species stored for 38 years at 5 degrees C and then transferred to minus 18 degrees C for about 25 years is only more than 100 years only for 61 of the species. So it's only 22% of the species retain more than 100 years of half-life. The rest of the species showing decline very quickly. And another report by Propert et al. Also showing that 26% of collection from 46 families showing significant drop in viability during 20 years storage under seed bank condition. We know that cell shrinkage beyond a certain percentage is lethal. And once the cytoplasm solidifies, cell shrinkage continues to occur, but over an extremely protracted time frame. So this can happen over the long time frame, and sometimes that may be related to the aging seen in dry orthodox seeds. So the effect may not be immediate, might be seen after 5, 10, or 20 years of storage. So just another note here that usually a lot of reports has been conducted on accelerated aging or artificial aging or controlled deterioration aging and environments. So this is usually to predict how long seed will survive in seed bank environment, but the actual experiment is conducted in high humidity environment and high temperature environment. So basically in a stressful environment. But caution is needed when interpreting the data and relating that to the actual seed longevity in dry storage, as the molecular mechanism in dry cold storage are very, very different from that of the artificial aging environment. So now it's widely recognized or accepted that accelerated aging and controlled deterioration conditions do not mimic the actual process of aging under long-term cold storage. So you can get more detail from our publication. And these are the two diagrams that explaining what the actual mechanism that's happening in the dry environment as well as the humid environment. So we talked about orthodox seeds storage, and some of the orthodox or desiccation tolerant seeds also will have oil content, and this oil may affect the storage of the seeds. And so when looking at oily seed storage, there are different factors we need to take into account. For example, what is the lipid content, the fatty acid profile of the seeds, the lipid thermal behavior, and then the lipid body size. Will these affect the storage of the seeds? So going back to the cytoplasmic solidification of oily seeds, it's a similar diagram, but some species have been reported that they've stored not very well under seed bank environment. And these species are, for example, Coffea, Carica papaya, coffee, citrus, and Salix and Populus. So looking back into the diagram here, now we have to have take into consideration of the lipid bodies in the cells. So what will happen when the cytoplasmic solidification happens? And that's in combination with drying as well as storing in cold temperature. So the lipid profile or the lipid bodies and the thermal properties of the lipid bodies are crucial here. Cells that survive drying may be damaged in the freezer when storage lipids crystallize and leave large gaps in the glassy matrix and increase the potential for oxidative damage or structural collapse. Cells that survive sufficient drying to approach cytoplasmic solidification but die with further drying. So precaution is needed when drying oily seeds. So going back to the water absorption isotherm that I showed for the 2% lipid before, now when we're looking at high oil content seeds, how does that change? So will that behave the same? So we looked at data from these seed information database from the Royal Botanic Garden Kew, and we looked at the relationship between seed water content and relative humidity in seeds. That is for 20 degrees C with different lipid contents. So the dashed line here represents water contents, as you stated, with 20% relative humidity for different seeds that contain different lipid content. So for P, that has got 2% lipid content, the water content will be about 6.8% at 20% relative humidity. For lettuce, that has got 30% lipid content, the water content will be around 5%. And for peanut, which has got 60% lipid, the water content that they will equilibrate will be around 2.9% at 20% relative humidity. So we know that now that seed with different lipid contents will equilibrate to different water content at the same relative humidity. This is critical information for any gene banks because usually we have a drying environment set up at one condition. For example, at Kew the seed bank, we have 50% relative humidity walk-in dry room that we put all the seeds there for drying. But we know that this will not work for the high lipid content seeds because they will equilibrate to much lower water content, which could be damaging for storage of the seeds. So just to show that how some oilseeds may not store well at minus 20 degrees C, we compared data for brassica seeds that's been stored for 40 years, and these seeds were stored, the same species were compared from Kew seed bank as well as the seed bank from Madrid, the plant germplasm bank in Madrid. So we looked at the seeds and we could see that some species are sensitive at minus 20 degrees C, or they're short-lived at minus 20 degrees C compared to other species. So we looked at the lipid thermal profile of this species. The long-lived species showed that they contain lipids with much lower melting and temperatures. So we looked at the thermal profile using the DSE, and we noticed that the long-lived species have much stable thermal profile, lipid thermal profile, and the lipids are melting at lower end compared to those seeds that stored or poorly stored at minus 20 tended to have lipids with crystallization and melting transitions spread over a wide range of temperature and also span over the storage temperature at -20 or 5 degrees C. We concluded that the seeds tolerate long-term cool, dry storage best when the seeds have stable thermal property, and when they're so they will be at the low risk of storage. The poor storage could be also associated with the presence of metastable lipid phase at temperature they are being stored. So, for example, if you're storing at -20 degrees C and the lipids are at mix of crystalline and liquid phase, that might cause a problem in storage. And we also did another study comparing 18 species of Asteraceae storing at -20 degrees C and -196 degrees C. This is the cryopreservation temperature. And we found there are two groups in these families. One is short-lived and one is long-lived at -20. So we found that at least 5 species are short-lived at -20. So when we look at the thermal profile, the lipid thermal profile of these species, comparing short-lived and the long-lived species, we found that the short-lived species not necessarily have high oil content, but they do have high lipid melt temperature. And especially for the peak 3 that highlighted here, all the short-lived species do present or have this melting peaks at high temperature. So now we're looking at the fatty acid chain length, oil body size, to see if there's any difference between these short and long-lived species. The other species that we looked at for storage is Actinidia species or kiwifruit species. These species are desiccation tolerant, but they have high oil content. So our previous study looking at 7 species storing at 3 different temperatures at 5 degrees C, -20 degrees C, and cryopreservation showed that some species show decline after storage 1 year in -20 and 5 degrees C, and some of them even after 2 years, there's a decline in germination. And most of them do well in -196 or cryopreservation temperature. So we looked at the lipid thermal profile again using the DSC to see if there's any indication of the lipid instability. So as I mentioned, kiwifruit or seeds do have high oil content, and the DSC thermal profile revealed that the lipids are in transitional phase or mixture of liquid and solid phase at -20 degrees C. So this is the thermal profile, -20 degrees C, and we can see the lipids are still melting at that temperature or the storage temperature. So we're suggesting that lipid thermal fingerprints can be used for rapidly identifying potentially short-lived oily seeds under dry cold storage. So we can use this as a biophysical marker for predicting seed longevity. So as a conclusion for recommendation of storage for orthodox and short-lived or intermediate species, for short-term to medium-term, we can still use fridge or freezer temperature of 5 degrees C or -20 degrees C respectively. But if you're looking at for longer-term, cryopreservation is recommended. So for cryopreservation of seeds, there are different factors that need to be considered or critical factors. One is seed maturity. As I say, it's very critical. The seed will attain full desiccation tolerant when they achieve full maturity. If you harvest before full maturity, they might show high sensitivity to desiccation. Seed moisture content is critical. Optimum moisture content must be attained, and ideally a glassy state is recommended for cryo storage. Low or high moisture content will create problem. Cooling and warming rate is also important, and that will affect survival after cryo. And physical damage to seeds. This is usually depending on the size of the seeds, because the seeds are capable of expanding and contracting with such cooling and rewarming rate. So it's really critical to see the seed size is amenable for cryo. Too large seeds may not be tolerate this extreme exposure to temperature. So we may have to look at other sources for cryopreservation or other material. For the whole seed cryopreservation can only be applied for orthodox and intermediate seeds. For recalcitrant seeds or desiccation-sensitive seeds, usually zygotic embryos will be used for cryopreservation. So we need to excise the embryos, and then we need to go through that same desiccation procedures. Challenge of particular significance for cryopreservation recalcitrant seeds that produce large embryos. So sometimes we even have to work with the embryonic axis instead of the whole embryos. So when we look at these axes, so the post-cryo recovery can be again challenging, because we may have to include in vitro manipulation or tissue culture to regenerate these embryos. So one example here is a cycas, which has got really large embryos. This is too big for cryopreservation. So usually we just slice the embryos to the axis, and we use the axis for cryopreservation. So different cryopreservation protocols can be applied. I'm not going into detail for each of these techniques, but they all work on the same principles, that we need to dry the seeds either by direct drying or indirect drying. And sometimes we need to add cryoprotectants for protecting the seeds, for preventing ice crystallization during cryopreservation. Sometimes we occur the problem where there's an embryo tissue heterogeneity, different embryo sizes and shapes. Chemical composition of the embryos may influence the uptake of the cryoprotectants. And so we need to optimize the protocol based on this. And sometimes the physiological status of the seeds as well, or the embryos, will affect what protocol that we can use for cryopreservation or cryoprotectants. So one innovative technique that we did was to apply vacuum infiltration to facilitate fast and uniform permeation of cryoprotectant solutions. So we tested this on 3 different species, and here we're reporting on Laurus nobilis. So the heterogeneity in morphology, physiology, and cellular chemistry in plant tissues can compromise the successful cryopreservation. So what we want to see is, can we facilitate the uniform permeation or the infiltration of the cryoprotectants using vacuum? So the vacuum infiltration cryopreservation techniques, we found that we can offer, or it can facilitate tenfold uptake of the PBS2, and the time is much more shorter, and the plants produced after cryopreservation was much more vigorous. So, and the cryoprotectant can be applied at 0 or 25 degrees C. So this is one advantage, because shorter application means like the plants are exposed to toxic cryoprotectants for shorter period. That's producing much more vigorous plantlets after cryopreservation. But also for tropical species, exposing cryoprotectants at 0 degrees C usually will be very stressful. So we can apply this technique at 25 degrees C, which will be widely applicable for tropical species. So the vacuum infiltration vitrification cryopreservation may apply a basis for more generic high throughput technology for cryobanks. And sometimes what we found is a generic cryopreservation protocol may not work for all species. We need to have a personalized cryopreservation method. So we know that applying cryoprotectants is critical to achieve the glassy state, but how much cryoprotectant need to be applied, or how much cryoprotectant was uptaken by the tissue is sometimes not known. So we can use biophysical techniques, for example, using the DSC to see how much we can quantify the cryoprotectants that's been permeated or taken by the tissues. Here I've shown the example of the corica papaya that we applied cryoprotectants for different time frames. So the dashed line is the control, and then 30 minutes, 60 minutes, 90 minutes application of cryoprotectants. In this case, we used plant vitrification solution 2 as cryoprotectants. And then we noticed that around minus 40 degrees C, that is where the PBS2 solution solidifies. So we can quantify that, the amount taken by that plant tissue at that temperature for different treatments. So when we plotted that against the regrowth of the plants after cryopreservation and the enthalpy produced by the DSC, we found that the internal concentration of PBS2 optimum for regrowth was around 67% for these embryos. So having known this, we don't have to treat the embryos with 100% concentration of the PBS2, but we can actually reduce the concentration of PBS2, and we can treat the embryos for longer period at this concentration because we know it's not going to be toxic for these plants. So this is another way of like how we can personalize the cryoprotectant treatments for treating a particular species that's a very difficult to conserve using any other conventional methods or generic cryopreservation method. So there are many advancements in cryobiotechnology, cryoengineering that help us understand how cryopreserved samples in much more efficient way. So biophysical studies ensure the understanding of glassy state that can be achieved and sustained. Thermal analysis using DSC and dynamic mechanical analyzer can be used to identify glasses, and programmable freezers can be used for stepwise freezing. Cryomicroscopy can be used to localize ice crystallization in a cell. So if the desiccation has been achieved or glass transition has been achieved, or we can completely avoid ice crystallization. As I mentioned, personalized cryoprotectant treatments are becoming also gaining attention now for difficult to cryopreserve plant materials. And people are also looking at rapid freezing in flush nitrogen temperature, which is at minus 207 degrees C instead of liquid nitrogen temperature at minus 196 to allow for rapid freezing. So coming to the last point of the talk, some take-home messages from this. What we have learned so far, we know that seed aging is influenced by external and internal factors that could be measured through physiological, cytological, and genetic changes. Increasing understanding of factors induced cytoplasmic solidification and affect glassy properties. And we know that cytoplasmic solidification slows down, but they do not stop the chemical reaction involved in aging. So aging will still continue to happen. Chromosome damage and gene mutation increase with storage time and inversely correlated with post-storage germination. And increases in oxygen tension, temperature, humidity were already known to accelerate aging, but it was the gradual accumulation of toxic metabolites that was considered the main causes of aging. And then we also know that degradation of proteins, lipids, and nucleic acids damage cell constituents and reduce seeds' metabolic capability, eventually impairing ability to germinate. Before ending the talk, I just want to say that we are not at the end of this discovery. The discovery is still happening. More research has been conducting and new areas are emerging. For example, in seed storage, the biochemical changes in seeds during storage has been studied, and there are several other areas where oxidative stress, RNA integrity in seeds during dry storage, seeds dormancy in storage, seed microbiome in storage, and cryobiology and biophysics. So there's more areas are developing to understand seed storage science better. So in conclusion, Seed storage biology is a complex science covering seed physiology, biophysics, biochemistry, and multiomic technologies. Simultaneous knowledge advancement in this area is necessary to improve seed storage efficacy of crops and wild species biodiversity conservation. And creating knowledge is just one part of the story. Translation of the science into practice is what required. So there are many ways this can be done by using the technology transfer platforms, technical guidelines, workshops on different techniques, webinars, training videos, discussion forums, and etc. Many organizations are already doing this. This list here is not exclusive, and you can check on their website. There's so many informations there available. So for most of the time, we don't have to reinvent the wheel. Some acknowledgements to all my collaborating institutes. And many thank you for your attention.
Thank you very much, Jayanthi. This was a wonderful presentation. Very interesting to hear new information on cytoplasmic solidification, lipid melt, and glassy states. So thank you very much for that. We have a couple of questions, and we'd actually like to go through them if we can. The first one is, do you recommend the use of vacuum when people have foil packets, aluminum foil packets? Given what you've talked about, the environment and the relative humidity and moisture content, do you recommend vacuum packing or just doing the regular packing with the aluminum foils?
For storage, usually we don't use vacuum packing because the effect of oxygen is really critical. So usually we try to eliminate as much as oxygen. Is usually in seed banking, we say drying is the first, so moisture content is the first thing that we need to look at, and then temperature. But sometimes a role of oxygen is forgotten, and that's why the vacuum packing came in. But for longevity, it's best to not to vacuum pack. So dry the seeds and keep them in the airtight container, but it doesn't need to be vacuum packed. But for short-term storage, it's fine. But for long-term storage, probably is not the best practice.
Okay,
from
good.
my understanding.
Yes, that is really good to know, because we have two camps, I believe, and I think the standards say one thing, but we're actually doing something else. So it's good to know your point of view on that, and we will take that into consideration. We have another question in French. And the question is, are there any studies that can explain the part of the ecology and the physiology of the components of storage of grains? So that is my understanding of that question as I translate on the fly. So are there any studies that you know about that explain the ecology and
So I think they mentioned grains. I believe it's a domesticated species they're looking at, but maybe we can go back to the wild relatives. Yes. So where they come from originally, and that might affect. But the trade might change, because with domestication, sometimes the trade will change, and the desiccation sensitivity trade will change during the domestication period, because these species might have been domesticated for a few hundred years now. So the trade of the desiccation tolerant will change. But if you're looking back, if you want to trace back, please look at the wild relatives of these species. That might give you some clues.
Okay, very good. We have another comment. Firstly, I wanted to say that this was a great presentation and very engaging. Thank you. My question is, can you explain a little more on personalized cryopreservation? You mentioned that some species require very specific conditions, and can you maybe elaborate a little bit on that?
Yeah, so personalized cryopreservation is derived from the concept personalized medicine in humans. So this is like more customized cryoprotectants or cryoprotecting treatments that we can apply to plant material they're working on. And this is especially for the plant materials that are very difficult to cryopreserve using the conventional or traditional methods. So more resources need to be investigated or put into this. Usually looking at applying the cryoprotectant, because cryoprotectants are quite stressful for the plants. So applying the cryoprotectants at the right concentration at the right time is crucial. And this can be affected by different factors: the size of the explants or the embryos we are cryopreserving, the dimension, whether it's a flat explant or is a cylindrical or spatulated embryo. So that will affect the uptake of the cryoprotectants. So we need to see what is the best timing, and sometimes the stress can happen even before we apply the cryoprotectant. So we can assess the viability at every step to see where we can optimize this protocol. And this concept has been introduced by Kim and his colleagues. I think it was published in Trial Letters 2012. So you can look back into the literature, and he compared applying cryoprotectants at progressive concentration for different crops. So we not necessarily need to apply 100% cryoprotectants because that's very toxic. So we can actually go for even 60% cryoprotectants might be enough for some species. So that will give us some flexibility to handle the material differently and less stressful for that plant material. So this is what we call as personalized. Yeah. It takes more investment in time and resources, as I mentioned. So ideally, it should be focused on maybe those are the critical to conserve or highly risk of extinction or highly threatened species.
Wonderful. Yep. So personalizing that concentration versus the time it's exposed, you might be able to get something that's optimal for a particular species. Yeah. Okay, well, I don't see any more questions. So with that, first of all, I'd like to thank you for a wonderful presentation. We did get a lot of people, despite it being a strange hour for Europe and Africa. But thank you very much, Jayanthi, for preparing this presentation for us and putting us up to date. And obviously to the attendees, thank you very much for attending today and widening our discussion with your questions. Any last words as goodbye, Jayanthi? That's over to you then.
Thank you, Anya. I just want to say thank you very much for this invitation to speak. It's a great honor to be part of the GROW webinar. And thank you everyone for joining this talk and for those great questions. Thank you and take care.
Thank you, everybody. Bye-bye. Thanks again, Jayanthi. Bye.